Hi guys, so today we'll be talking about leftover analysis. Hi little guy. Wait, and there. Leftover analysis today. Okay guys, so in leftover analysis, what happens is that you usually have a different reference build like HG19, and you're trying to convert to HG38. So, which is HG19 is also like the HG37, and you're trying to convert to a different reference build. So how do we map from these different genomic coordinate systems? Liftover. Hey, little guy, let me kind of show you how that is possible. So there are different genomic coordinates out there. So for instance, if I'm looking up this study on bipolar disorder, so it's a Genome-Wide Association Study that is from 2019 for bipolar disorder. So I navigate to the folder here, and I can see, the first 5 rows, for instance. So it's like chromosome, SNP, base pair, A1, A2. These are all the columns here, the headings. And so this is a SNP here. And the first one is chromosome number, then SNP ID, then base pair. But we don't often know like what is the reference build. So there are many different reference builds out there. For instance, if you go to this caviar or any site in general, it's going to say human reference version freeze is. So which version is this? HG19, which is GR37, or HG38? And they seem very, very confusing. And this is something I also didn't really know. It's not something you might think about often, but these are different mappings. So if we were to look, for instance, at this example here, let's just look at this bipolar disorder. So, so in other videos, I've talked about how you can view the first few rows of the Genome-Wide Association Study, because there are like so many different lines in this. 2019. You can see that there are just so many rows in this file. So, about 13,413,245 rows, including the header. So let's see that, okay, this is chromosome 10 and position 239426. So the following information that we can gather is that this is chromosome 10, base pair. So chromosome number and then base pair. So we have CHR10 and the base pair is 2392426. So that's the first one we can look at. And the next one is just the next row, which is, so that's red. Okay. So the next row is, so this one is corresponding to this over here. So this row here. Let's see, this is corresponding to this. And then we can also just do the next one, which is just chromosome 10 again, and this position 2855051. And then we can also take the corresponding SNP IDs, which is RS, this is the SNP ID here, and then this is the SNP ID here. So for the next one. So let's just see what happens. So what we are doing then is I'm sort of taking that and in this information here, and I just sort of put it in here. So I took the chromosome 10. I took the chromosome 10 and then this position here, which is what you see, here. So I just took this first row of information and I put it here. And then I also put the corresponding RSID right below it as well. And then I also took chromosome 10 and this one here, and I put it here in this next row along with the corresponding SNP ID below. So I want to show you what happens when we try to map it, because I don't know, like, as of now, I wouldn't know like what is the reference build. So I'm going to start with HG19. And what we'll see is that this returns CHR10 2392426. This, maps here. this 2392426, yep, this maps, and the RSIDs that were picked up is RS185339560, which actually is the same. So that's great. And similarly, chromosome 10 and this position 2855051, it's great. And the SNP ID of RS80145673 maps. That's awesome. And I can assume that that would probably, be the case going on, just for those two. But why can I assume that? And why is this really huge, that it's an HG19, they map properly for HG19. What we are trying out is HG19, GRCH37. And I have another video where I kind of walk through in more depth how to use this site to map, genetic variants and do those conversions. So HG38, let's try it now on HG38 and see using HG38 what we get. So does it all work out? Well, okay, we'll find the SNPs here, the SNP 18. Okay, so we can look up on RS185339560 in HG38 instead. And what we can see is that this SNP is good, okay? And similarly, the SNP RS80145673, okay, the SNPs are mapping. Now let's see about the positions. So this SNP here, the position that was picked up for it is 2350232. Does that actually map here? Does this position actually map with this? It doesn't. It doesn't map. This is different. This is not the same. If we were trying to see if there's a map from here to here, that is definitely not the case. It's not mapping properly. And then in the next example, what we will see is similarly, if we were to look at this SNP here, RS80145673, which one does it map to? This one here. Let me change the color. Let me just make this orange. Okay, so if we look at this one here, does 2812859, does it map? No, it does not map. It's not the same. It's not. And in fact, what you're going to see is that it sort of says that if it were to look up, chromosome 10, position 2392426, if it were to look this position up, which is what we want, it says it's not found in HG38, but it's found in HG19. And similarly, this position here, CHR102855, this position here, 55051 is not found in this, but it's found in HG19. So what that goes to show me is that what we show here is that the same SNPs can be detected in HG19 and HG38. For the most part, I think they can be detected, but the positions that they correspond to could differ. So while these SNPs are the same, these two SNPs are the same here and here, they map, the positions do not map and they're not found in this HG38. So what this goes to show us, is instead, it goes to show us that in reality, the build is probably HG19, because these positions, this CHR10239, this is a proper map. This part here is a proper map. This and this is a proper map for the same SNP. So that's great. It's a great map. It's great. It's awesome. We're happy. That's great. And same in this other situation with this position. So we see that the SNPs are the same. And, for HG19, it even told us that HG38 even told us that, hey, you probably are going to find that in HG19. So what you're going to see is that the same SNP can have different genomic coordinates. So one build is very different from another. And there's this term that we need if you need to map between different data files of different types. One file may be in HG19. So guys, just to note, so we have HG19, which is the same thing as GRCH37, and HG38, which is the same thing as GRCH38. So it's kind of weird why this is the same and why this is kind of different, but these are the different builds that there are out there. So how do we map them exactly? So there's this concept that is called Liftover. This tool converts genome coordinates and genome annotation files between assemblies. So HG19 is a different assembly from HG18. So basically, so this is UCSC, so Genomic Visualization and Interpretations. To motivate that, I want to say is that a common analysis task is to convert genomic coordinates between different assemblies. Probably the most common situation is that you have some coordinates for a particular version of a reference genome, and you want to determine the corresponding coordinates on a different version of the reference genome for that species. For example, you have a bed file, that's one of the input files, with exon coordinates for human build GRCH37, which is HG19, and wish to update to GRCH38, which is HG38. So what you're wanting to do is you may want to convert from the build GRCH, you can write it either way, 37, which is HG19. You may want to convert it to another file where it's instead the type of coordinates. So GRCH38, which is HG38. So I've shown to you that the same SNP can have different coordinates because it's like a different mapping system. It's like a translation that you need to do. So you need to do the mapping between them. So you might need to map in some cases this way. You might need to map it in this direction in some applications. But in other applications, you may need to go the other way. If you're going in this way, the conversion would be HG19 to HG38. You would need to do this conversion if that's the direction you're going in. But if you're going the other direction and you're trying to map it, going in this direction, then what you would need to do is you would need to convert from HG38 to HG19. So you would need to do this conversion here if you want to go in that direction. So this was sort of inspired, and this is a very helpful site. So let's kind of go over, what is a reference assembly? And not just this, we also have reference builds and sequence alignments between humans and canines, which is what this is trying to do, canine family, like the dog family. We even have it for cows, for different types of mapping. If you want to see on our chromosome, on our DNA, what is the coordinate system and where do certain regions tend to match or align properly? Like how do you change or adapt the coordinates exactly? And that's important because some of the Genome-Wide Association studies, some of the other files that are working with single nucleotide polymorphisms or genetic regions, they can be using different assemblies. So I'm going to talk to you about it and also how you can deal with that. So let's go over what a reference assembly actually is. It's as complete as much as possible representation of the nucleotide sequence of a representative genome for a specific species. So here we're focusing on Homo sapiens, HG. Is what it is. So there are a powerful shortcut when mapping reads, as they can be mapped to the assembly rather than to each other to piece the genome of a new individual together. This has a number of benefits, the most obvious of which is that it's far more efficient than attempting to build a genome from scratch. By its very nature, however, using this approach means that there's no perfect reference assembly for an individual due to polymorphisms like SNPs. This leads to the publication of new, so it's due to the presence of repetitive structural elements like duplications, inverted repeats, tandem repeats. A given assembly is almost always incomplete and is constantly being improved on. So this leads to the publication of the new assembly versions every so often, such as GRCH37, which is 2009, GRCH38, which is December 2013 for the Human Genome Project. It's also important, so every few years when data is getting better, they try to update it routinely. So like GRCH37 from NCBI, so different organizations can publish different reference assemblies. So this is coming from the NCBI, while HG19 is coming from UCSC. They are identical, save for a few minor differences, such as in the mitochondrial sequence and naming of chromosome 1 versus CHR1. So there are some resources available to convert coordinates from one assembly to another. So I'm going to talk about like how you can basically access, a chain file from UCSC, but use this R function and Bioconductor package called RTrackLayer to do the mappings across assemblies. And this is important because of what I showed you, where when you try looking up in HG38, the certain positions, they're not found. A SNP with that location is not found, but in this file. So what this goes to show me is that the bipolar disorder, this is instead, it's HG19 data. And let's say I want to convert, I want to go and I want to convert these coordinates here. I want to figure out for all these base pairs, I want to convert each of these to the HG38 build. I want to go through and convert. I want to go through and convert each of these. How do I go about doing that? That's what I'm interested in, is like going and converting it. Because as you see here, what it's saying is that this information is not found in HG38, but in HG19. So this is HG19. This is HG19 data here. HG19. Which is the same thing as GRCH 37. So we need to convert this. That's what we need to do. So I'm also going to motivate it here, where it says that the conversion rates from HG38 to HG19 average 95% were lower than the conversion rates here. Then they talk about a number of significant single nucleotide variants that the same thing as SNPs could not be converted between this and this. We recommend the newer version, which is HG38. So it came out like, 4 years later for a next generation sequencing single nucleotide variant analysis. So our findings suggest caution when translating single nucleotide variants between different versions of the human reference genome. So we even see what that means. So based on what I thought a SNP is, if I had gone with, if I looked these up in HG19, the coordinates that I get here are very different than what I would get if instead I look these up using, so basically, if I look these up using HG38, these SNPs have different coordinates. You see this? And this is very different from the same SNP, this and this, that's the same. So between the builds, this is HG19, HG38, the same exact build is there. This is the same. But what's different is that these are different. These are not the same. And similarly in the other situation as well, where, CHR 2855051 for this is very different from what you get. This value here is different from this part here, that they're not the same, even for the same SNP. So you have to be very careful. So let's see, how do we go about doing this, given that the build is different, HG38 versus HG19. So let's go about doing that now. So what this site was talking about is it was talking about doing UCSC Liftover analysis as well. So I go here to the website here. So this is, the UCSC, University of California's website here. And I can go, so it's hgdownload.soe.ucsc.edu.downloads. So I can see this, and what we're trying to do, our goal here is to go from, for the bipolar bipolar disorder, one of the brain-related disorders, that affects mood, stability. So for bipolar disorder, we're trying to go from HG19. So this is the 2019 GWAS, Genome-Wide Study. We're trying to go from the HG19 all the way to, which is the same thing as GRCH37, to HG38. So whatever the purpose is that we need to do this conversion, that's what I'm trying to show you, how we can do this. So that's what our goal is, trying to do. So yeah, just a little spiel on bipolar disorder. So it's still important because it's a disorder associated with episodes of mood swings ranging from depressive lows to manic highs. Exact cause is not known, but a combination of genetics, environment, and altered brain structure and chemistry may play a role. High energy, reduced need for sleep, and loss of touch with reality. And depressive episodes may include symptoms such as low energy, low motivation, and loss of interest in daily activities. Mood episodes last for days to months at a time and may also be associated with suicidal thoughts. And treatment is usually lifelong and often requires a combination of medications and psychotherapy. So it's very, very, devastating. So manic phase may last weeks or months, and depressive phase could also last that long too. So just to bring some awareness as well about what this is. And there's a Genome-Wide Association study on it that was actually published that we're using here. It's published in Nature and a really good paper. So it's highly heritable. And they did a Genome-Wide Association study with 20,352 cases and 31,358 controls of European descent. Which is why in, when I read in this file here, what you're going to see is that I put in for cases and controls, I sort of put in, okay, 20,352 cases and for controls, 31358 controls. And this is the total sample size is 46,582 individuals from this study here. And that's when I'm reading in this SNP file because we're going to be using R. So when I read it in, what we're going to see is that this is sort of like reading in, we have the chromosome. Actually, let me also just show you view SNP file. While that is loading, I want to kind of show you what we do here. So we want to do the HG19 all the way to HG38. So we want to go from this to this build. So you can do all sorts of things. You can do all sorts of conversions. But so I'm going to go here. And I'm going to click Liftover. So Liftover is like kind of like translating. It's like a translator for translating the different genetic genomic coordinates. So I'm going to go to Liftover files. And then it's going to kind of tell me how I can download all the files if I want to. And this is like HG19 to all sorts of things. There's Bos Taurus. This is like a cow genome. So you can map a human to a cow genome, which sounds it's really awesome. And you can map different genomes together. So I want to do HG19 to HG38. This is what I want. And this is what was published on pretty much New Year's Day so like in December 31st, 2013 at like 11 or 8 p.m. They published this conversion file. So people really are into this science even on New Year's Day. So you can see, you can just download this .over.chain.gz file here. You can download it, and then you need Ubuntu here, because what you're going to do is you're going to navigate to your downloads folder. And then what you're going to do is you're just going to do gunzip HG19. I put it in quotes, to hg38.over.chain. I think maybe I downloaded that again twice. 1 second. Actually, I need to navigate to my downloads. So that's important. You need to be in the right folder. So then I'm just going to go back up and I'm just going to run this. And I'm just going to say yes, because I had done this earlier, before I'm showing this to you guys. So now all you have to do is do gunzip. So what you're going to see then, is you're going to actually end up seeing this. You're going to end up seeing this hg19 to hg38.over.chain. So it's a chain file. So why do you need that? Well, we're going to need it to load it into R to do the conversions. So what the formula is that I typically use, not the formula, the package, I mean. It comes from RTrackLayer. So RTrackLayer is a package available on Bioconductor. And along with many things that it does is it allows you to change coordinate systems using RTrackLayer Liftover. So before I get into that again, I kind of want to show you that basically whatever you want to download, you can just download it. Like you can even hg19 to hg18 over, you can do all these things. But what this file, if you click hg19, it's going to show going from hg19 to other reference builds, So if instead we wanted to do the other way around, if we wanted to go back and instead go from, we would choose this if, like, let's say, so we were looking up before hg19 to hg38, but we would choose hg38 to hg19. In fact, they even show this example here, that if you want to go from hg38 to hg19, then you go there. And then you could just download that file and that's going to help you go the other way instead. But since we have hg19 and we want to go to hg38, that's why we downloaded this file over here so that we could convert our Bipolar Disorder Genome-Wide Association Study results. And the results from this study were kind of cool and very informative. So this is what the Genome-Wide Association Study results look like. These are SNPs, single nucleotide polymorphism. So Genome-Wide Association Studies, negative log 10 p-value, and then the chromosome for all SNPs, for all single nucleotide polymorphisms. Also known as variants. SNPs or variants, just SNPs. So the important thing is that like it's just a single nucleotide, so one DNA base, ATGC, being different in at least 1% of the population. So it's quite frequent. So it's a variation. So this is the Genome-Wide Association Study, and the build happens to just be hg19. So that's why we downloaded it. So RTrackLayer is the package that we will need in Bioconductor. So if you don't have it, you're going to need to download that. You're going to need to download RTrackLayer in Bioconductor. And you're going to need to input the chain. So let me kind of show you in the context of, the bipolar disorder example here. So this is the bipolar disorder. This is, like all these different rows, 13,413,244 entries, the chromosome, the SNP, the base pair, A1, A2, other information, odds ratio, standard error, p-value, which is important, and other things that you may need here as needed. So these are summary statistics that are essentially when you see basically just to kind of show you guys again, that you see the chromosome, the SNP, the position, each of these dots is a single nucleotide polymorphism. And you take the p-value here from this column, and then for these chromosome numbers, and these are the base pairs, these are sort of like, you take the negative log 10 of this p-value, and that gives you your y value, and then you just continue plotting these dots and then seeing what reaches genome-wide significance. which is usually 5 times 10 to the negative 8. So you can do that. And that's sort of like what that data looks like again, but we want to convert these coordinates. So all our goal is to convert these coordinates here for the same, we want to, chromosome will be the same, but just like coordinate, we want to update, go from that's our goal. So I've loaded it in here. And all our goal is it's just to update this column of base pairs because again, like we want to really update. We really want, to go from HG19, we want to go all the way. So again, as you see here that this is the same, 239, 2426, that's the same, for this SNP here. And this one, the second row, 10, this part is the same for this here. So just showing you that what we were looking at before was just the first 2 rows here. That's what these were here. And I just want, and this was for and we now know it's HG19, that's what we found out. And we just sort of want to update it so that we can, find the corresponding. So ideally, what we would want is we would want this to just say, hey, in HG, we would want to replace this top one with 2350232 instead, because that's the updated coordinate in HG38. So that's what we would want to do. We would want to instead like kind of, replace what we have here with this coordinate. So what you will see, let me show it this way, is that we have, we have this RSID 185339560. Make it my signature, my favorite Comic Sans. Eeks. So what you're going to see is that we have like, okay, so we have this over here. Hopefully I got it down, but if not, you see that this is just the same what I'm trying to get at. And what we will see is if we go down this path here, now let me make it more like a tree and let's size it bigger and like this. So if we go down HG19, which is what it actually is, then for this SNP, and this is on, we know that it's on chromosome 10. So we know this, but what we're trying to do is we're just saying, okay, well, if I go down this path, I know that the position is going to be 239, 2426. And indeed, like this is also important stuff, this is like sort of like what a chromosome, each chromosome, this is like chromosome 10 here, chromosome 10 in a karyotype. And actually it has about so many different base pairs. If you look at chromosome 10, you can see that it has quite a lot of base pairs, tumorigenicity, megaloblastic anemia, severe combined immunodeficiency disease. So people trying to do all these mapping, obesity, medulloblastoma, all these different things, so each of these mapping, so we're trying to map from one coordinate to the next. And what we'll see here is that this on chromosome 10, actually in HG, so if we look here at HG 38, it's actually, oh gosh, I have to use this, 1 second. So what this actually ends up being is, it is 235.0.232. And what our goal is we just kind of want to go from this to this. For this SNP, we just sort of want to update this with this value. We want to just stick this value in, we want to take this value and put it in. That's the conversion essentially that we're trying to do. Just get that coordinate because the SNP is the same. So the SNP is the same. Chromosome 10, they both map to the same SNPs. You see the same SNP here, it maps to the same SNP. So what we want to do is we just kind of want to update the position number. We want to go from here to here. We want to switch this with this. Okay, so hopefully that makes more sense to you guys. Okay, so then the next thing that we're going to do is we're going to go back here to the leftover analysis file. So I have read in the cases controls, I viewed what it looks like. Then now what we need to do is we also need to call this library. Let's call in. So if you don't have this in Bioconductor, this is an R, so biomanager.install, and this is called RTrackLayer. Okay, so I have it installed. So I'm just, once you have it installed, you just call this RTrackLayer, and then you load it in. Okay, great. So now I've loaded this in. It took me a while, so I've already run all these lines here. As we see above, I've sort of run all these lines here, reading it in, and this is, I just write the names, case controls, and then they also have the NCA and NCO. They also have those, so I just have an N sum where I'm adding those up together. So what we need to do is we also need to, so what we had done, remember, is that we sort of had unzipped this file. So now it was a .chain file that we had shown you guys. Remember, it's a .chain file here. So part of the requirements for RTrackLayer is that you need to load in. You sort of need to load or import the chain for a path that has your chain data. So in this example, I'm doing HG38 to HG19, transformation to HG19 coordinates. It's like, a different coordinate system, like different year, different, as they're going from British, date, which is like day, month, year, to like American, which is month, day, year. So it's like you're just having to do some conversion. So similarly, what we have to do is we just have to, once we have this package RTrackLayer loaded in, we see where it is that this file is. It's in my C drive, my users, Saniya, downloads, and this is where it is. So I'm going to call this object to import. I have to import this file here, import the chain file. So please import it. So this is the chain object here. So it's a chain. Okay, and what we see is that what's really important is that these chromosome numbers begin with CHR. In our SNPs file, our chromosome numbers do not begin in CHR. So that's going to be very important. We need to add in, CHR in front of our chromosome numbers. We need to add that in, going down for all of them. So what I'll do is I'm just going to add that in, I'm just going to create another variable for SNPs file. So I'm going to just say SNPs file, new chromosome column is equal to paste 0. So this is how you can set each and every single row in a column to a value. So paste 0, like if I can do paste 0 for any variables, like let's say A equals 5 and B equals 3, then paste 0, A is A and B is, and this will allow me to paste them together if I want to add in an extra, hey, I forgot to add in an extra of this. So now you can see that this sort of allows you to take a variable, which is 5, which is what this is. Take a string, whatever you want, you can paste elements together. You can even do paste, A, B, and the regular paste adds a space in, paste 0 does not add a space in. I somehow just like paste 0 because that makes me feel like I have more control. because you might add an extra space and, and not remember that it adds a space. So paste 0. So I'm going to see what is the name of my column that has a chromosome and SNPs file. Well, it is called SNPs file, CHR. And then I want to just add, in front of that, I want to add CHR in front of it. So CHR directly in front, no spaces. Because I wanted to match the same format as my chain object. I didn't do that at first, so I'd gotten some mistake. And then I'm like, hey, why is that the case? So CHR, that's what we want this mapping to be, the CHR here, so that it'll map to this too. So otherwise, when you do it, it's not going to work without that. So it's going to take, it's doing it for like 14 million. Oh, it finished very quickly. So I can just do head of SNPs file, just do the, please see the first 5 rows. And you'll see that indeed what we got is what we wanted right now. We got like CHR in front of everything here. So that's like really good here. So you have CHR here, that's really good. So then the next thing that you're going to want to do is then you're going to see that this is a new mapping here that takes place. So the next thing that you have to do, I also do this because I want to keep track of it, so you can do SNPs file. I can just say old HG19 base pair, or you can call it BP. So BP is not blood pressure here. BP stands for base pair. So what is the base pair location? So this SNP, single nucleotide polymorphism, is at chromosome 10, position 239, 2426. So I'm just going to set this equal to SNPs file. And this will really depend on your Genome-Wide Association study, but this is just to remind ourselves that this base pair was HG 19. So if you just want a reminder, what is this exactly, then it's just a reminder for you to tell you that, hey, so once we run this, I'll show you again. All that's doing is it's just creating a duplicate row just so that you can keep track of it. So you can see here, this is these two are the same. I just kind of like to keep track. This is optional for you guys. You don't have to do this, but I just wanted to keep track because you might see BP and you'll get very confused because the coordinates are like pretty much, there's no really easy way to tell that, that this is HG19, like what is what. It's not really easy to tell that this is HG19, this is HG30, and that we're trying to go from here to here. There's no very easy way to go about doing that. So that's why we just have to have the column name so that later on when you're reviewing everything, you're like, okay, yeah, I see which one was the old one, this is the new one. So just don't confuse yourself, that's all. So that's what we need. And then we also need this G ranges object to be made. But before we do that, so we have a new chromosome column, which is what we need. This is what we need this. We have this. Then we also kind of need to incorporate the start field, which is this. This can also be the start field, or we can just say SNPs file start field is equal to, we can also just set this here again. We're just creating another duplicate. This is just the start position for the SNP, and SNPs file end field is also the same thing because the width of the SNP is just 1. It starts and stops at 1 DNA base. So it's always this base pair here. Start and stop is just this base pair. It's just the same thing here. The end position is this start position because SNP width is just 1. It's just 1. 1 DNA base. That's all it is 1 DNA base, single nucleotide, single, 1 base. And then what we'll see here is that we have all these additional columns that we should keep, which is great. So I'm just going to do this where I'm going to just say genome. So this is genomic ranges that I'm going to be doing. Genomic ranges is G ranges. So I'm just going to do GR, GWAS, SNPs. This is I'm creating a genomic ranges object from a data frame, because the structure right now for SNPs file is that it's a data frame. So I want to convert it to a genomic ranges, because it's much easier to do the liftover analysis for the two genomic ranges, because this makes it so much easier to overlap like two huge files. Because you see, like, this is already like, you know, almost 13.5 million rows. So that's a lot. So there's this formula here that's called make G ranges from data frame. So inside that, I want to put in SNPs file. This is the data frame that I want to convert to G ranges. Then the next thing I'm going to do is have seq names.field, which I'm going to say is, you have to put it in quotes. And what that is it's called new chromosome column. So this seq names in the G ranges object is just the name of the chromosome. That's just with the column that has the chromosome information. So name of column with chromosome information for the SNP. Or anything, but in this example, it's a SNP. Then I also need to create the start field, which I'm going to say, what is the name of the start field? Well, I just called it start field. That's the name of the column here, start field. So in quotes, you just have to say, like, what is the name? Name of column with start.field. So this is the start of the chromosome of SNP position. And in here, the start is the same as the end, but we just sort of need to do it because G ranges, like, it's not really explicit way to say that, hey, this is the same, you know, start and end. So end field is equal to end field here. So you're going to just say this is the end dot field, end of SNP. And this is the same thing, you know, these are the same. If it's easy, you could also just have done this. It's equivalent because they're exactly the same. It's up to you. The width is just 1. But if they have more than 1, then what would it be? Then, and that's why I want to show you the end field. You know, because of what if you have like different, like if you have ranges, like enhancers, promoters, distal regions, other things, then how would you do that? And then there's just one more argument, which is keep dot extra columns. And I'm saying true. Why? Because I don't just want to have my my chromosome column start and end. That's not all I just don't want to have. So basically, let me sort of show you once I add this and this, let me show you. Okay, so essentially I'm telling it to make it based on this here and this part here. And it's going to know, it's going to figure out that, hey, the width is actually 1, they're the same. It's the width is 1. But I just don't want to have just these 3 pieces of information into it. Oh, before I forget, I should also I also want to add this in here before I forget. SNPs file source equals, just so that we remember. So we want to have all the other information too. We want all these other columns, even if some of them may be redundant, we want to still keep these. We want to have the SNPs and we want to have the alleles, the frequency of each alleles, odds ratio. We want to just keep that information right now. So what I'm going to do is that's why we say keep extra columns is true, because we don't just want these 3. We don't just want this column and this. We also want like the source and all this other information. So we're just going to say keep extra columns true. Might take a little bit of time to make this object, but it's going to be very, very efficient. And then the next thing that we're going to do after that is, yeah, let's just see what it did. Like GR, GWAS, SNPs. I put an extra G here. So yeah, now what we see is that, we have the essential, which is seq names, ranges, and strands. So this is all that we would have had if we had said false to this. If we said false, then we would have just had those columns. One more thing I want to tell you, because you might forget about this, is this new chromosome column, guys. This new chromosome column was this one. But what if instead we put CHR instead? Like, what if I just forgot to update it? We did the updating by having the space 0 CHR. What if we forgot about it and just put CHR in? We wanted to see how, but actually I'll do that after. So I'm just going to show you. So this is our GWAS SNPs. So I'm going to show you this one. I'm going to show you this one is a bad one. And I'm going to put in the CHR here and then show you what happens. So it's not going to complain right now, but I'm just going to show you that it's going to probably not work. I learned from experience that it doesn't work. So then I'm like, wait, why isn't this working? And this is my first time running this package. Okay, so we can try it the bad way as well, where we just don't label the number and we don't put CHR in front. But the one that works is going to have the CHR in front of it. So the next thing that and you can see it's genomic ranges. So it has all this information and it's a G ranges object with everything here and all the metadata columns. It's really nice. So then the thing we're going to do is we're going to then just run Liftover. So I'm going to start with the bad example first, and I'm just going to show you that it's not going to work. See, discarded unchained sequences. That's what happens, is that it just doesn't even know what to do. So it's like, I don't know. It's like, this is returning 0. And then you'll be like, wait, why? Why isn't it working? And then you might come to the conclusion that, hey, this actually uses chromosome names like this. So if you instead try to do it with, numbers instead of the CHR, then the number. So it really depends on the format of the file that you get. So this does not work. So instead, we're going to need to use the one with CHR in front of it. We're going to need to use this format where the seq names is instead CHR10 instead of just the numbers. This bad one. This bad one was a baddie. So it did not work for us. So instead, what we're going to do is we're just going to try this with this GWAS. I just want to show it just because you might forget, and I want to show you kind of what happens. And it's just not me trying to show you a bunch of rules. You might have a little panic like me. I'm like, why isn't this working? And like, am I doing something wrong? Am I breaking it? And then I'm like, oh, wait, I forgot CHR. I didn't really think it would be that sensitive. So this hopefully should be the liftover analysis that you get. And you see here, I have this old HG19 base pair, so we can then just see what we'll end up getting afterwards now. So now, okay, great. There was no error. There was nothing like this coming. So I was so happy at the time too. I'm still happy. Okay, great. So now we see, okay, seq names is this, and because we kept the original, we still have this original column here. I know a lot is redundant, and because these are huge files, you might want to remove redundant things. But now what you're going to see is like, okay, this is the same. And if you didn't kind of save the old one, I mean, we have it here, which is 239, but you might be confused. So all you need to know is that, okay, this is the old base pair for this first row. The old one was these end up being the same, but I remember BP is not sometimes as informative. So the old one is 2392426, which is what we were looking at here. 2392426. That's the same one here. And remember that we were wanting it to be 2350232. We're converting from HG19 all the way to this. We wanted it to be this. And is that what we get? Do we get 2350232? Well, yes, in fact, we do. We get that. That's awesome. So it did the mapping correctly, you know, it did that. So this is the start and end, are the new coordinates that we are getting here. That's what we wanted. This is the same new coordinate that we were wanting to get. That's awesome. So in essence, you can just, you can get rid of end and start. It's just keeping it there just because you gave start and end. And you might have regions where maybe you have at least 1,000 or not at least 1,000. You could have many base pairs, so the start and end could be different. So what I'm going to say here is results. New HG38. BP is just results, you know, start. And then I'm just going to say head of results. So I can see we see first 5 rows. And then I can also do dim of results is to see the dimensions of results. And it kept the original columns, which is great. So now we have all this information, group 1, you know, we may have to drop that afterwards, but we have what we want. We have the mapping. And just to also check here. So if you recall, we also had tried this out for caviar. We had tried out like that, you know, HG38 for this SNP 8014673. It should be 28. We said that the position, remember the corresponding position, it could not find. So let me put it this way, just to, you know, show this other example too, is that this position here was the old one for the second row. So guys, just remember that, you know, we're looking in the second row here. So when you're looking in our output, this is first, second, third, fourth, fifth, sixth rows. But what happens is that just spilling over. So, you know, this is a continuation of the same row. There's so many columns. So this is the second row, which is corresponding to this SNP, corresponding to this SNP, you know, 8014. And what we see here is, let me just move that up here and just try that one this again. This one is this here. And the one, oh, sorry. This is HG38. What it had said is that this, okay. For the same SNP here, sometimes I confuse myself. I'm like showing different coordinates and I get mixed up too. This part here is like this here, that's the same SNP, so holding a constant. And this is the old, that was not found in HG38, but it was found in HG19. That's what we had tried putting in before for that SNP. And the new one that they recommended for us was actually, they recommended this to us. And when they recommended this value to us, this is just the same as this. It's just the same thing. That's exactly what we wanted, and that's what we got. This is exactly the same. So this is an example of how this mapping has worked on multiple, these 2 different examples. So it's just allowed us essentially to just do the conversions. And I do recommend, you know, having this here and having this part here. So then you can also see, you know, the same, the start is, or the end is just the same. So you can see the conversion of coordinates. So now we can kind of work with either we have what we want. We have, for a given set of SNPs, we have the old HG base pair information. We have the new HG information, and this is just a head, the first few rows. And then I think 5 or 6 rows head shows. Might be confusing Python with that. And then we also have, you know, the SNP IDs. As well as I think this is like a deletion. This D stands for deletion. And that just shows how we can do this mapping, and not just on chromosome 10, but on all the chromosomes. Yay, yay, yay. So that is just an example here of how we can do this mapping. have for a given SNP, we have the old HG19 base pair. Now we have the new HG38 base pair. So we have this conversion that we did here, and that's the best thing. So that is just, the best thing that we did. We did that conversion. So just to revise with you guys again, just very quickly, is that we just we load in a genome-wide study. We just have to also load in the chain file, which we downloaded, and gunzip, G-U-N, gunzip it. So once you download it, you just do gunzip. So you go here to just download it. I think you go, let me see. You go to UCSC site here. Yeah, you can go here to downloads. Under downloads, you can check out, the browsers, and then you can see the human genome, and then you go for whichever one you want to go to. So again, this is going from HG38 to any other reference genome, even for cats, dogs, cows, anything. And this one is going from HG19 to any other reference genome. And again, HG19 is the same thing as GRCH37. And this is just the same thing as GRCH38. So that's why you can go to any of them, and you just have to click on the leftover files here or here as needed, and then download the file that you need. From here, the conversion file. You download it, and when you downloaded it, all you have to do is just gunzip it. Like go where it is and do gunzip. So make sure you have Ubuntu. I have videos on downloading that as well. So then you can just gunzip it. And then after that, you have to run this code in R where, once you load it in, you have to make sure you add a CHR in front of your column so that you end up using, so you're going to end up using this data here. And it actually disappears after. So you see how the chromosome, it takes that information on the start field and end field and the new chromosome column, and they're no longer here actually. That's why I was saying to try to store the old base pair information as well, like try to store it because that might be gone. You see like all these columns that we essentially had used here, these column headings, they kind of disappear in the final output. So sometimes it's just good to keep track of the old column. So if you're doing HG38 to HG19, if you want to save this as HG38 base pair, and I put old to represent that that's what we had before, and this is the new. So, you just create the G range, it's some data frame. You need your chain object again, and your chain object is just you importing, please import the chain file. And this is just whichever you want it to be. HG19 to a cow, a horse, a mouse, the mouse genome as well. It can be anything. So you don't want this. This one is not good. You want to get rid of that. This one is a baddie. It has a CHR information, not like numbers. Oh, my star ability has gone down. Ability to draw stars. Anyways, so you kind of don't want this. You want this part here, your SNPs file, your sequence names field, the name of the column with the chromosome information, the name of the column with the start field, name of the column with the end field, and then this could be the same for a SNP. This can just be exactly the same. It's just telling it how to do it. Keep extra columns is true if you don't want to lose this other information. And then what you do is you just run Liftover. This is the Liftover function again. The Liftover, and then you put in your SNPs file object here. This just comes in. This object comes here. That's all. Like this object comes in here. Oh, I don't. Got red. Nice and cheerful. This comes here, and then the chain object has whichever HG file to whatever. like chain file. So that's sort of how this does the conversions. And then in the end, we have what we wanted, and that's great, so yay, we just finished doing the conversion. So that's sort of how I got by with trying to do the conversion, and then still kind of working on that. And I just found out about how to do this conversion. So I'm just sharing it with you guys to help you guys out as well if you have similar difficulties. So how do you change genomic coordinates? How do you do that? And that I hope that this helped you guys in the process of doing that. Please let me know if you have any questions, and subscribe if you liked this as well. So thank you guys, and I'm Saniya, and hope that this was helpful.